Hello students, welcome. Now this time I'm going to start with the new topic, that is structure of chromosomes. So students, as you know that all living organisms on this earth are made up of cells, and inside the cell, inside the nucleus of the cell, there is a genetic material. That genetic material contains all information about that individual, either that is plant, animal, insects, and any other living organism, microscopic or multicellular. They all are having cells, and inside the cell nucleus is present. Inside the nucleus there is a genetic material. That genetic material is called as chromatin fiber also, and that chromatin fibers are also known as chromosomes. So what is the relation between this all chromatin fiber chromosomes that we are going to discuss in this video? So first of all, what are chromosomes? So students, as you know that the genetic material is present in the nucleus in normal condition. Whenever the cell is in resting phase, that genetic material is called as chromatin fiber, because it is a very thin coiled chromatin, coiled fibers, that is called as chromatin fiber. And when there is a cell division start, at that time this chromatin fiber just get condensed, and the nucleus, which has so much chromatin fiber, that chromatin fiber become condensed and converted into a chromosome. And you can see here the chromosomes picture is given. This chromosome is a highly coiled and condensed chromatin fiber, and in the middle there is a centromere. Actually that chromosomes become duplicated, and that forms two sister chromatids, and these sister chromatids are joined at the centromere, and then they will form a chromosome, highly coiled condensed chromatin fiber. And chromosomes are actually, why they are called as chromosomes? Actually chromosomes readily pick up certain dyes. Some dyes they can pick up and get colored, hence the name chromosome is given. And chroma means colored and soma means body. So you can see here the colored chromosome. Then after that, the chromosomes when start condensing, then they will get duplicated chromosome is shown here. Centromere is in between and showing two sister chromatids are there, and these sister chromatids, you can see highly coiled structure is present. Highly coiled chromatin fiber is just condensed, get collected, and if you will take out one thread, that is a chromatin fiber, you can see here. Okay, and this is a condensed and duplicated chromosome. Now you can see here that when these chromatin fibers are further observed, then they will look like a beaded structures. You can see here this is a beaded string is visible. Original picture is this, and beaded on a beads on a string. So similarly you can see here there are some thread like structures which are coiled around the green structure, that is beaded structure is there, which is coiled and highly coiled and form a chromatin fiber. Now I am going to discuss this in detail. Chromatin material actually made up of two substances, that is DNA, deoxyribonucleic acid, which is 40%, and histones, a type of protein which is about 60%. So these two when combined together, then they will form a beaded structure, that is called as nucleosome. These beads are called nucleosome. You can see here this bluish H2B1, H2B, H2A, H4, H3, H1 also there. These are histone proteins. You can see here histone proteins, and this coiled structure, this thread like structure is DNA. It is coiled over the histone protein. These histone proteins are 8 in number. They will make a one group, and that is called as octamer of histone. 8 histone proteins when combined together they form octamer, and that octamer is coiled by a small piece of DNA, and that then it will form a one nucleosome. DNA strand wound around a core of 8 histone proteins, that is called as nucleosome. And afterwards they will be look like this structure. You can see here, these are the nucleosomes. When they join together, then they will just become highly coiled, and then they will form a chromatin fiber. When chromatin fiber get condensed, then they will form a duplicated chromosome. Okay, this genetic material is in the form of chromatin fiber, and when the cell will undergo in a cell division, then this will form a chromosome. Okay, so this is about the chromosome. And now I want to tell you about discovery of DNA. Ultimately, students, the chromosome is made up of DNA and histone protein. So DNA is actual material of which the chromosomes are made up of. So DNA was first discovered by James Watson and Francis Crick, with their model of DNA is presenting here, and they actually presented this model of DNA, and after that they got Nobel Prize in 1962. Along with this, James Watson and Francis Crick, they have prepared the molecular structure, but they were not having idea that of which shape and structure is it. And after that they have taken help of Rosalind Franklin, who got the X-ray diffraction pattern of the atomic structures of the molecules. So this is a DNA atomic structure, that is a X-ray diffraction pattern. From this technique only, Watson and Crick got the idea of exact spiral and double helical structure of DNA. So by the help of this technique, they presented the DNA model. So DNA was discovered by Watson and Crick, okay, in 1953, and they got Nobel Prize in 1962. This is all about structure of DNA. Rest we will discuss in next. Thank you.